computer, share the screen, pick up our PowerPoint, share. All right. All right, so this is coming for everybody. So I don't see, yeah, there's Preston. Sorry, Preston. Yeah, this material is coming in micro. So this kind of gives us a way of introducing the fungi to you in an immunological way, but it also gives you a little different take. Some of the tests, and what you're going to find out is we don't have just a whole lot of tests. We do have some, but a lot of times it's actually just collect, stain, and look. So in the back half of micro lab, you will have slides to identify. So we won't have samples we will have some plates growing of these. And we may try some of the things like putting them under the scope and staining. But at the end of the day in micro, you will have to identify slides. So there'll be a slideshow and you will identify the different funguses. Okay, so based on their look. So we do that toward the end, but this is kind of our immunology way of getting it started. So if you don't know what a fungus is, it's a heterogeneous group of eukaryotic organisms. And eukaryotic is different from what we've been talking about, right? We've been talking about prokaryotic for most of the way. So what is a eukaryotic? What changes? What makes a eukaryotic a eukaryotic? They have a nucleus, nucleated. Is that it? Yeah.
I don't know.
So, I mean, y'all had this since what?
Huh?
Huh?
Like one had like a cell wall, one doesn't.
One has a cell wall, one doesn't. Yeah,
Yeah, I
but,
can only remember like small.
yeah, but which one?
Oh, which one's which? Oh.
Yeah, which one?
Doesn't pro, that's before the nucleus.
Pro before the nucleus. They don't have a nucleus. Prokaryotes. Didn't y'all have this like sixth grade? Sixth grade science, like didn't they go through eukaryotics versus prokaryotics? What made them different? Help me out, Zoom. Putting you to work. Your classmates.
They have a nuclei and rigid cell walls.
Who has that?
The eukaryotic cells.
The eukaryotes have a nucleus. They're cells, right? And have a cell wall. What did you say? Okay. What's prokaryote?
I assume none of that.
Assuming it doesn't get us where we want to be. What's prokaryote?
They don't have a nucleus, but they have a single piece of cellular double-stranded DNA located in the area of the cell.
Okay, so nucleus is our key. Okay. Or do they have, yeah, they have cell wall, right?
I don't know the difference.
Didn't they? Don't our bacteria have cell walls? That's what we stain, right? Stain cell wall. Gram stain. The nucleus. Okay. So the nuclear material would be in a single area instead of scattered throughout. Right? Okay. Y'all with me, right? All right. So inside the fungi group, right, we have distinction. We have what we know as yeast and what? We have two. We can split these into two. What's I guess what is different about fungi? What can they do? They can exist two different ways. Okay, some of them are dimorphic. Some of them can be yeast, and then they can turn over and form spores. Okay, so mycelia fungi, mycelia, there's the third little list on our, see our PowerPoint? Isn't it up there? Everybody see that? It says mycelia. So the yeast are unicellular forms of fungi, and mycelia are multicellular forms, and they produce through spores, production of spores, CANDIDA. Okay, so we have some that can do both of those. Like they may grow as a yeast, like we may grow them up when we get to this point in micro, we may grow them up as a yeast overnight. You look at them, well, look at that, yeast are growing. Okay, but they may also possess a spore forming form, okay, a mycelia form, where they turn in not just into a single form of a yeast budding out to the next one, but they can actually have multi-cells connected to them as they grow and they reproduce by spores. So that's key to kind of our fungal direction, where we're going. Don't want you to miss that. That they can be dimorphic. They can, some can go from yeast, right? And we think of that with urinary tract, and we're looking at urines and we say, oh yeah, there's budding yeast, yes. But then we may have a skin scraping and we may see this weird looking thing that's got spore heads on it, like a head comes up and starts spore producing, and that spore is kind of the problem we have with them in immunology and with our patients. So that's when we kind of pause to get ready for fungus. All right. So if it is an infection, as you see, mycoses is an infection caused by fungi. They can also cause hypersensitivity, MYCOTOCCUS, mycetium, tissue infections. So we have issues, and your book goes into really, y'all studied this, right? Because y'all had all weekend, y'all should have already read through this chapter. That's kind of where I'm digging for that. Right? You took a quiz, and you had the chapter, you already knew this chapter was on the test. Anyway, so what is the problem with our patients with fungi? Who's going to be our main concern? Which patient? The young, healthy? Are you worried about fungus infections? You worried about getting introduced to a fungus that happens to be on the shower floor growing? No? No? Are you worried about eating fungus in your food? Something growing on your cheese? No? No? Not worried about eating that? Do any of you have fungus growing in your refrigerator right now? Any of you have fungus growing on your shower floor right now? Any of you have fungus growing on your feet right now? Go there. Anybody? Anybody making homemade beer at home right now? Go there. Right? Anybody? No? Okay, so who are we worried about? We're not worried about us. Who are we worried about? You have read the chapter. You don't have to be ashamed about reading the chapter. You can let people know you've read the chapter. Immunocompromised patient. Yes, that's all this section is about, is our immunocompromised patient, right? Because that's who's going to have these. Normally, these are just kind of everywhere, and we're not really worried about them because they're usually superficial, right? And unless we go into this time of year, I know I've maybe mentioned this before, but my old hometown haunted house had to be closed down because of bird guano. Anybody know what that is? Bird poop? You ever heard of anything being closed down because the birds are nesting and roosting in an area and pooping everywhere, and all of a sudden you can't go in there because maybe one of these that we're going to talk about today has been detected there. All right, so we had that concern, or I did back as a kid in my house, my haunted houses. So all of a sudden, you know, we kind of go through this. I love how well, I don't love this, but the PowerPoints here just kind of, you know, jump pretty quick through things. But let's look. So tissue infection, there's 4 different types of mycosis. And a mycosis is a fungal infection. You didn't catch that. Okay, so if you got an infection or disease caused by a fungus, you have a mycosis. If it's plural, as it is on this slide, it's mycoses. So these are infections involving tissue infection. So we know the superficial, right? We know the athlete's feet. Fungal are, and these are cosmetic, they're irritating. Then we may go to cutaneous, and these are even worse, right? When our nails get fungal infections. Has anybody ever had a nail that's gotten infected? Like a toenail got infected, fingernail got infected. You know, we go to these little places around town that do our nails, right? And you can get infected while you're there because they didn't really take care of their stuff after they used it on the person before they start hammering it on your nails, and then you end up with a fungal infection. Nobody's ever had a fungal infection? No? It turns your nails kind of off color, looks gross, ugly. Y'all are lucky, y'all are very lucky. Nobody's had that? Nobody's picked up any Zoom, anybody? Nobody's gonna admit to it, right? So, you know, you can buy stuff and you put it on there, but I was gonna give you a homemade remedy in case y'all had suffered from one of these. And it is, Vicks Vapor Rub. You ever heard of that stuff? You put that on your nail with a fungus, okay? Covers it, knocks it out, knocks out the air to it, kind of smothers it out. It works, it worked on mine. Not that y'all care about what my feet looked like at one time, but yeah. So yeah, that was given to me by a doctor. He said, hey, try Vicks Vapor Rub as a treatment, and actually it works. And when you get one of these, they're long lasting, and they're like, ooh, they're irritating and they're gross. And you definitely don't want to come out of winter and put on, you know, go to flip-flops in the spring and have issues. So trying to help you there. Transmitted through direct contact with a person or an animal. So I know you've heard about athlete's feet, right? And showering. How many of you have gone to a community shower and wore your flip-flops while you were there? Like you didn't even want to take them off. You didn't want your feet touching the surface of the shower in the dorm. That was mine, or at Boys State, or wherever I was, right? Supposed to take those. If you just never know. So they can be superficial, just on your skin. They can be cutaneous, where they kind of move into our skin and nails. And then we have subcutaneous. These are a little deeper. Ulcerative skin lesions, subcutaneous tissue. Maybe we've had a trauma. We've gotten an injury while we were out, car wreck, going down the trail. We had an injury and interject environment into the wound. And then we may have systemic, where we have spread through the body. And a lot of the ones we're going to talk about today are systemic because they end up starting in the lungs. So we're inhaling them, and then that allows them to move into our body once they're inhaled. So kind of get you in the mood to talk about mycosis. So for us, it's always important that we want to talk about the AIDS patient, right? Does anybody know an AIDS patient?
I'm so sorry, my roommate locked herself out of my house. I'm joining Zoom.
Okay, so you're going to Zoom on the way out?
And yeah, she's locked out of my house.
Okay. All right. So has anybody ever encountered an AIDS patient? No? Would you know? Yeah. Would you know? Because see, the scary thing about HIV is that your age group is the young, increasing patients. Okay. All the new cases are coming from your age group. Teenagers, around teenagers to 20s. The other age, the other homosexual males population's decreasing their numbers. They're like the lowest, right? Which is a flip. So it's education. So it's safe sex education kind of thing in the growing numbers. So when I was teaching, we had high school, before I came here, we had high school students that were coming to class. And the reason I ask is because there wasn't one in my class that didn't know somebody had HIV. And there might be one or two at the school, but they all knew. They all knew who they were, and they all knew they were under 18. Okay, so that was the scary part, right? Because we're not all tested, we all don't know, and we may be encountering those people in our life, and we don't know that they are infected. So it's real scary to think about HIV at this point. I don't know if you guys feel that way, but that's kind of the word I'm trying to get to you. Okay, so the AIDS patient population is the one we're mainly concerned, but we're also concerned with the immunocompromised, ones that are taking immunosuppressive therapy because of autoimmunes that we've already talked about, our cancer patients, right, that are on autoimmune, chemotherapy, things that are destroying the immune system. These are the patients that we have to be concerned with the most as far as coming up with a fungal infection. So let's look at how the immune system, your expertise of how the immune system reacts to fungi. So we have our innate defenses. These are pretty simple. We know that, right? Skin and mucous membranes. We have our recognition system, if you want to call it the system, the PAMPs and the PRRs. And PAMPs are what?
Pathogen associated molecular
Patterns. Yes, good. And they're conserved among microbial species. The PRRs are what?
pathogen or pattern recognition. It's something recognition receptor.
Pattern recognition receptors. Our PAMPs are our pathogen-associated molecular patterns. So these play in the innate immune response with the cells, right? Our phagocytes, our macrophages, and our dendritic cells. So we have a response, right? It's recognized. The good thing about fungus, the reason you don't worry about it as much as other immunocompromised patients, is that the fungus doesn't have much evasion, doesn't have much to fire back with. So once they're recognized as foreign, your body pretty much gets rid of them for you, or isolates them onto the skin, and you hope that your immune system would help. But we've all, well, y'all, it sounds like y'all have ever had a fungal infection in your life. That's very, very good. So when you do get one of these frustrating ones, hopefully you'll remember this lecture. So we have four PRRs, and they say there's several classes, and they can either be transmembrane proteins, located on the cell surface, cytoplasmic. There's 4 different classes. Toll-like receptors, TLRs, our C-type lectin receptors, our CLRs. We have one called retinoic acid inducible gene, our RIG1-like receptors, our RLRs, and our NOD-like receptors, our NLRs, which are cytoplasmic proteins. It says, go back to chapter 3. Y'all been to chapter 3, but if you'd like to go back and review our innate response. Okay? We also have cytokine production, not cytotoxin, cytokine production and inflammation. And if you've ever had, nobody's ever had if you ever had a fungal infection, they're very itchy, right? If you ever see somebody with one foot above the other one, scratching their toes through their shoes, more than likely they have a fungal infection on their foot, right? Their feet or toes. And they're real, and still nobody wants them. Like right in between your toes. If you ever spread your toes out and they're like right in here, right in this part, like that's the most irritating place ever for these infections, right? Nobody's had athlete's feet? Okay, good for you. That's very good. Anyway, just to give you an idea, some of you maybe, the next time you see somebody scratching their foot through their shoe, you're gonna think, I remember Mr. Rector saying, I know what they have. They have a fungal infection on their foot. Okay, so after the innate defense, we also have active immune responses. Okay, your book goes into, the TLRs, and we went on through here where we have a lot more information about the TLRs and CLRs and how fungal recognition occurs. Then we get into, they say, okay, there's a couple of organisms here, CRYPTOCOCCUS and our histoplasmosis. Okay, this is when we start getting into kind of the cell-mediated adaptive immune response. So cell-mediated immunity, T cells and cytokine production are most important. And it talks about how there has been a debate whether we do have cell-mediated and humoral. It's pretty much cell-mediated these days, okay? You got chemokines, recruiting the T cells to the area of infection. You already know this. And the response to the fungi are specifically those mediated by T helper 1s and T helper 2s. T helper 1s have been shown in mice to be vitally important in the clearance of the organism, the ones we just talked about, CRYPTOCOCCUS and histoplasmosis. And then once a T cell has been committed to the response of the fungi, they express an effector function. They release interferon alpha, tumor necrosis factor alpha, and interleukin 17 and 22. And thus we get a protective immunity to pathogenic funguses. Okay. So how do we diagnose? Isolation of the organisms from the clinical sample. I think we've already talked about that. It is the gold standard, but it's very slow growing, okay? We're gonna see some of our fungi that are just gonna be like, boom, there they are. Look at them, look how good they're growing, because we're gonna supply some extra stuff for the growth that's gonna help them grow a little faster. Histological, that's staining. So we're going to be able to recognize when you get through with micro, the end of this semester, you're going to be able to recognize these as a picture. So if you do have them on a slide and stain them, you can see it. Skin testing. This is kind of be like what we do with TB, right? It's still kind of the same thing. Look for sensitivity to the spores of the fungi. We can do some serological detection, whether it be with our test kits that we've got for antibodies and antigens. And then there's molecular assays, which are just kind of getting started. So there's not, as it says, they're not widely available yet for use. So when I think about that, I think about a respiratory sputum panel for BioFire. That's the one we used to run. Also for blood. So we have these, and I can't remember the breakdown, but I'm pretty sure there was one or two on the list that were for fungal. Pretty sure for those two. Because a lot of times that's where we detect a fungal infection, is in the blood or the sputum, if it's gone systemic. That is, it has invaded the body and has moved to those areas. So when we think about antimicrobial treatment, okay, knocking out bacteria but allowing yeast to grow, we can see that sometimes with that scenario in the hospitals. Wound infection, open wound getting infected, because these are everywhere. We're not really concerned about them. We're not thinking about them being introduced with some of our equipment that we have in the lab, in the hospitals. So really does a good job going through that. There's one, I can't remember if it was, no, it's coming up. There's some testing. Yeah, I want to talk about complement fixation, but it's toward the end of the lecture. I couldn't remember if it was here where I wanted to talk about it or later. I think it's later, it comes up. So your book kind of gives you an intro to fungal pathogens, which I thought was really nice. Gives you a group of 5 that we need to worry about. There's going to be more than 5 on your little slideshow test that we're going to do. But this is a good start. I like these. So we got Aspergillus, we got Candida, we got Cryptococcus, Histoplasma, and Coccidioides. Coccidioides, Histoplasma, Cryptococcus, Candida, and Aspergillus. Sorry, not Gillius. I guess it could be Gillius. Aspergillus species. So I love when we start with the first 2. The reason I just put species out there is so that you can name all the others. All right, and there's a whole little list of Aspergillus. So Aspergillus, did we get a little, oh yeah, we get a little thing here. Good. So Aspergillus species, we get common species, Fumigatus, Flavus, and Niger. Okay, so the A in front is Aspergillus, Fumigatus, Flavus, Niger. So that's our ones, our common species that we'll focus on for now. We have some others, a whole list in your book, but we'll stay with these. These are pretty much ubiquitous, meaning they're found worldwide. There's over 185 species. They're recovered from soil and plant material. Oh, and if we didn't make point that these usually feed on stuff, right? That's how they get their food. So they're kind of considered parasitic, but they kind of sometimes just feed on dead stuff. So it's not like they're really killing the organism. They actually use the dead organism to survive. So we think about soil and plant material. It does have spores. Okay, so that's where the CANDIDA is known as the spore. And they may be recovered from indoor air environments. Oh goodness, all right, how would we do that? We have a little playful culturing from an air conditioner unit earlier in the semester between Cadence and myself. So yes, air conditioning units, all right? Vents, anywhere there's moisture, anywhere there's, you know, things that look like something's changing and growing on side of it, inside, you know, all that good stuff. So we can definitely find them. Fumigatus is the first one, is the most commonly isolated species. That's why it gets the first one up there at the top. Flavus and Niger being the other frequently recovered. So that's why we go with those 3. So we've already talked about how the immunocompetent host, it's, you know, if we are immunocompetent, that's us, right? Now, I think all of us are for the most part. External ear infections. We can get pulmonary aspergillioma, where we actually breathe in the species or the fungi, or then it kind of walls off into the lung. We can actually have allergic reactions to the spores. So we get an allergic bronchopulmonary aspergillosis, ABPA. So if you see that abbreviation, that's allergic bronchopulmonary aspergillosis. So that's us, right? We're handling that. We can still pick these up. We can still have problems, but definitely not as bad as the immunocompromised patient. Immunocompromised host ends up with invasive pulmonary aspergillosis and disseminated aspergillosis. So that's the spread. So it's spreading from being inhaled and causing us to have big problems. So this is us. This is the Aspergillus, right? So this would be, is this the yeast form or the mycelia form? See, let's go back to that and see if we know. Which one, does that look like it's multicellular with spores? With a head on it that's got a bunch of spores that are about to go like a, what's the little, what's that one? What's that little thing that grows in the spring? And is that a dandelion? Right, you pick up the little fluffy white stalk and blow those all over, let the wind blow them, right? The bright yellow flower that nobody wants in their yard. Remember those? It's kind of what I think about when I see Aspergillus hyphae here. It's just like we just want to go, and that's all going to go everywhere, right? And that's kind of how the spores get spread. All right, those spores get spread. Once they're spread, ta-da, you get more growth. Okay, so positive tissue biopsy demonstrating this hyphae. Okay, so that stain of the spore head. We can also grow it, so we can get a positive Aspergillus culture, and then we can actually do a detection of the antigen in the serum with our testing, our immunology testing. That's kind of the first one. Let's see, oops, I hit 0 instead of the arrow. So the second one, Candida, and you see one of the leading causes of the infection, albicans there. This is yeast that may exist as a commensal organism in the human host, meaning not maybe causing a whole lot of issues. Maybe, what's commensalism? What is, remember we defined that. We have, what's the definition for commensal organism?
Where it benefits both.
Benefits both? Is that right? Everybody? Is that right, Zoom?
I think so.
Think so? Commensal. Oh, we'll go with that. Somebody said, tells me different.
Wait, is both the mutualistic?
I don't know. We had like 5, didn't we? Didn't we have like 5 of these that I said, hey, you're gonna need to know? We can pause. Zoom, y'all, Mary, you're trying to help? You're green, that's why I'm asking. It looks like you're unmuted trying to say something. Is my volume down again? I can't hear you. Can Zoom say something and make sure we're connected?
Can you hear me?
Yay! Okay, yeah.
Yay.
See you too. Or I see Mary. Kathy's talking. Okay.
Well, Mary's talking.
Hello.
Mary, I can't hear you now. She's unmuted. Try again. Y'all keep trying to talk. I don't know what's up with the system here.
Still
Everything's
here.
good on my end.
There we go. All right. Kathy wants to say something, or Mary? Who wants to say something?
Yeah, it's colonization of the host with parasite with no benefit or harm.
No benefit nor harm. Is that commensal though? Right?
Yes.
Mutualistic relationship.
Symbiosis.
Symbiosis.
Is where it benefits
Both.
the host and the parasite.
Okay. Sorry, Zoom. Y'all, evidently technical issues have kept you from contributing that information to us today. All right, y'all got to remember that. Okay. Found on the skin, GI tract, female urogenital or genital tract, Candida albicans. There's Mary again. Hey, Mary.
Okay.
The immunocompetent The one, that's us again, right? We got a good immune system. Diaper rash, vaginitis, female urinary tract infection, and oral thrush. Okay, I'm gonna mute Mary. Sorry, Mary. I'm getting feedback. All right, so that's normal. We see that. We see that diaper rash. That could be on babies and adults, right? Vaginitis, big reason, big cause. Female urogenital tract infection, oral thrush, right? And your book goes, hey, if you're an adult and you don't really know why you're getting oral thrush, which is a yeast infection in the mouth, you may want to get HIV tested. And I love how they do that. You may want to go get tested for that. But we see that with infants, right, with oral thrush, maybe following an antibiotic treatment. And we see that. And then they have to do the antifungal instead of antibiotics, because what they did was kind of knocked out the normal flora. The yeast kind of took over, and we ended up with a problem. The immunocompromised host, we get oral thrush. That's our HIV patient, or our chemo patient, or our immunosuppressed patient, or our steroid patient. Whatever way you want to say our immune system's being diminished. Esophagitis, inflammation of the esophagus, pneumonia, septicemia. So, so that sputum that comes to us, we may grow that up. It might be high bright, and it's not staph epi, but it might be a yeast, okay? So yeast can show itself on our blood agar plates from our sputum cultures, or even our urinary tract cultures, okay, depending. So it's always nice when you do a urinalysis, if you do see budding yeast, point it out. That'd be great, right? That way we kind of know, hey, this may have potential yeast growing in this culture that you're about to put on blood at McConkey, and you end up with a yeast infection. Okay. So recovering Candida, we would do a culture media. We can grow it. We can do a potassium hydroxide, is that it? Potassium hydroxide, wet prep, you might have heard it called that, of clinical material, looking for budding yeast. We can also do serological, where we're looking for the mannan and the anti-mannan antibodies. Colonization can stimulate antibody production in an uninfected person. And immunocompromised patients may not produce detectable antibodies. And that's what every section, when they said, hey, it's time to do an antibody test, then they said, but consider your immunocompromised may not have that ability. They might not have produced an antibody because their immune system's been suppressed. So even the antigen being there, the antibody test might not be a good thing for an immunocompromised patient. Then they talk about molecular assay, sensitive and specific for diagnosing early infection or whether it's an invaded infection. All right, so we're on to number 3 of our 5, CRYPTOCOCCUS. CRYPTOCOCCUS neoformans. They just didn't even worry with the other species. They just went straight to CRYPTOCOCCUS neoformans. This is the one, it's starting to become a problem, right? When we have bird guano, bird poop, okay? It's encapsulated, so this is an encapsulated yeast. So your book comes up with a really good picture right here of an India ink prep, and hopefully you can see the capsule around it. So you could see that real easy. And they say, yeah, these are easy to perform. They have a high specificity, but they have a low sensitivity. So let's go back. So they are contaminated bird poop, okay, is good. Enters host through the lungs, meaning, think about my haunted house as a kid. The birds are roosting in that all winter, and then they say, okay, we just have all this poop everywhere. All right, so think about barns, birds, in the barn. I think about my magnolia bush, because they're not trees. I didn't know if y'all knew that or not, but magnolia, there's no such thing as a magnolia tree. They're magnolia bushes. But that's where my bird problem has been, because they get pretty tall. They can get 20 feet tall too. But the birds in the winter about this time like that tree because they got those big leaves on them, kind of get in there, kind of makes a hut kind of feel. And then they like roost in there. So then there's all this bird poop at the bottom, right? If you've ever had a magnolia bush, you know that the leaves fall, and they're those big waxy leaves, so they're kind of hard to mow, and then they got all this poop underneath them. Anyway, so we can breathe that in, causing a range of manifestations from just maybe asymptomatic to a life-threatening pneumonia. So this is a problem that can get serious. It disseminates to the central nervous system, causes a meningitis or meningoencephalitis. And the majority of symptomatic infections are in the immunocompromised individual, which we started off with our AIDS patient. But you guys say, you know, don't know if you're ever going to see an AIDS patient. So we may see somebody that's just under immunosuppressant therapy because of their autoimmune disorders or their cancers, right? Taking chemo, those kind of things, radiation, bone marrow transplant, all those good things. So if we did our serological test, serum would be a great start. Cerebral spinal fluid, looking for the cryptococcal polysaccharide antigen. And we can do antigen titers, our latex agglutination. What is LAF? Remember what was LAF? Let me unmute all my people over here so y'all can hear. How many people do we have on Zoom?
Lateral flow immunochromographic assay.
Lateral flow. That's what we do, right? That's what we did the other day in lab. Did we do a lateral flow?
That was the strep.
Did strep? Yeah. I know, but it was kind of the same technology. Good. So there we are. Right here, picture. See that little strip right there? It looks familiar, right? It came in this little cup with a lid on it going up. Looks very familiar, doesn't it? And it's cryptococcal antigen test. You can actually have that. All right, so now we end up with our very last one. Is this the last one? No, we got two more, right? Yeah, we got two more. Histoplasma, CAPSULATUM, dimorphic, meaning what?
It can be both single and
Exactly, it can be a single yeast or it can be a multicellular mycelia. Found in the Ohio River Valley, Mississippi River Valleys. That means we're in that ballpark. Acquired by inhalation. I think this is the one that did shut down my haunted house back in the day, was histo. Acquired by inhalation of the mycelia fragments and spores. So definitely those spores come into play here. Mainly asymptomatic and self-limiting. Opportunistic infection with immunocompromised persons, individuals with AIDS. We can get an acute or chronic pulmonary symptoms, most common. And then again, it can disseminate to the organs rich in mononuclear phagocytes, fatal if untreated. So it's very treatable, but if you go without treating it, it can be fatal. So here we can culture histoplasma too. We can do serological tests for the antibodies, but complement fixation and precipitation are the most common. And we're going to spend our last little bit talking about complement fixation. Again, antibody to the histoplasma glycoprotein. That's less than 10% of the patients. Antibody to the M glycoprotein in most patients, but cannot distinguish between current and past infections. That's a problem. Skin testing comes positive 2 to 4 weeks after infection and remains positive for life. So if you did a skin test for histoplasma, you would know if you ever been what? Exposed to it. You actually do a urine test looking for the antigen too. All right, and our last one to finish up is our coccidioides immitis, dimorphic again, prevalent in southwestern United States. So we don't see this one as much, but it could still be around for us. So we inhalation of the CANDIDA that produce mild infections, similar to community-acquired pneumonia. Again, we can do serological testing, culture and identification in the fungus, immunodiffusion, and enzyme immunoassays, looking for IgM and IgG. All right, so what I wanted to touch on right at the end, see if I had, oops. So we'll quit the share, stop the share. We're just gonna finish, we got a minute. So what I wanted to point out to you on page 404 is the complement fixation. So complement fixation, look for antibodies using complement fixation assays. This is like the next to the last paragraph in 404. When mixed with coccidioidal antigen, an immune complex is formed with the patient's antibodies, resulting in a depletion of complement. When complement is depleted, antibody-coated red blood cells added to the mixture fail to lyse. Okay, so this is again, that's one of those where we talked about it in the lab, where you had the red blood cell not lysing was a what? Positive test, right? Yes. So if the antibody is there and complement fixation is looked for, it should lyse the red cell, right? If the antibody is there. The test is often referred to as an IgG test because IgG is the immunoglobulin class usually involved in the formation of this immune complex. CF, complement fixation antibodies, are detected later and for longer periods than 2 precipitin type antibodies. Okay, so that was what I wanted to point out, was how complement fixation worked. Is if the antibodies are present, the red cells are added, they're sensitized, so if the complement is there, right, and sensitized, it should lyse the red cell. So there'd be a titer that's considered positive. All right, we'll spend a little more time on that. I think we ran out. All right, we'll get back to complement fixation. Need to cover like a little bit more. All right, we're going to end. See you later, Zoom.